Hello everyone, in this class we'll learn about vestibular apparatus. The learning objectives of this class would be to name the components of vestibular apparatus and give their function, understand the mechanism of action potential generation in hair cells, understand the physiological significance of hair cell arrangement and their responses in otolith organs and semicircular canals. Trace the vestibular pathways, name the vestibular reflexes, understand the vestibular dysfunction and list the vestibular function tests. Vestibular apparatus is located in the bony labyrinthine of inner ear. Though the ear is popularly known as for its special sensory function of audition, it is equally important for its non-auditory sensory and motor functions, that is, maintenance of equilibrium at rest and balance during movement. The receptor for equilibrium and balance are hair cells that are located in vestibular apparatus. Vestibular apparatus consists of semicircular canals and otolith organs. They detect the sense of position and motion of the head. Movement of the head occurs with the movement of the body. Thus, the vestibular apparatus indirectly detects the position and movement of the body. Vestibular receptors are stimulated by linear and angular acceleration. Linear acceleration stimulates otolith organs and angular acceleration stimulates semicircular canals. They maintain equilibrium by activating postural and ocular reflexes. Vestibular apparatus is enclosed in the bony labyrinth of temporal bone. Vestibular apparatus is also known as membranous labyrinth. The membranous labyrinth contains endolymph. The endolymph contains high concentration of potassium, which is similar with that of intracellular fluid. The space between membranous labyrinth and bony labyrinth contains perilymph, which resembles extracellular fluid in its composition. So this is the structure of the membrane labyrinth, where it shows the cochlea, saccule, utricle, and three semicircular canals, anterior, posterior, and horizontal, which are situated 90 degrees to each other. The vestibular apparatus contains of otolith organs and three circular canals, semicircular canals. Otolith organ are referred to the two sacs-like structures called saccule and utricle. Saccule and utricle contain calcium carbonate crystals, crystals of the ear or otoliths, for which they are called as otolith organs. Saccule communicates with the cochlea via ductus reunions. Three semicircular canals on each side arranged in three mutually perpendicular planes. Accordingly, they are named as horizontal, anterior, and posterior canals respectively. Their arrangement ensures that the least any one of them of either ear can detect change in rotational acceleration in any plane. The hair cells are the receptors in vestibular apparatus. Hair cells have a large number of cilia arranged according to their length. The longest cilium is the kinocilium, which is located to the extreme end. Other cilia are called as stercocilia, which are arranged in a graded length with longest stercocilium remains next to the kinocilium. Hair cells have directional sensitivity. When cilia bend towards the kinocilium, hair cells are depolarized or stimulated, and when cilia bend away from the kinocilium, hair cells are hyperpolarized or inhibited. The hair cell activity is conveyed to the higher centers through 8th cranial nerve. Bending of the cilia towards kinocilium opens potassium channels through which calcium enters hair cells and produce depolarization. Bending of cilia away from kinocilium closes potassium channels and prevents entry of calcium that produce hyperpolarization. So this is the diagram showing the hair cell with the nucleus, and here the hair cells, the longest of which is called as stercocilium and the shorter one they are called as stercocelia. And this is when the hair cell, when it is not active, and when this is the hair cell when it is depolarized, and you can see increased discharge from the vestibular nerve, and this is when the cell is inhibited, that is hyperpolarized. You can see the number of reduced discharge from the hair cell. Otolith organ. The receptors in otolith organ are hair cells. Hair cells in saccule and utricle are located in the macula, the sensory epithelium. The macular hair cells are covered with an otolith membrane, which is gelatinous mass containing crystals of calcium carbonate, known as otoliths or otoconia. The cilia of the hair cells project into the gelatinous mass and are embedded in the otolith membrane. The macula of the saccule is oriented vertically and macula of the utricle is oriented horizontally. Hair cells of the otolith organ are stimulated by linear acceleration. Thus, the saccule detects linear acceleration in vertical direction and utricle detects linear acceleration in horizontal direction. They also detect change in the head position, like tilting or bending of the head. So this is how the hair cells are arranged with the supporting cells, and this is otolith, and you can see the hair are embedded in the otolith membrane, and this is the arrangement of otoliths or the hair cells in the utricle, and this is the where they are situated horizontally, whereas in the saccule the horizontal cells are placed vertically so that they can detect the vertical acceleration, whereas utricle they are going to detect the horizontal acceleration, linear acceleration. The cilia of the cells are embedded in otolith membrane that contains otoliths. Otoliths are heavier than endolymph. The specific gravity of the otolith membrane is more than that of the endolymph. Therefore, a change in the direction in the gravitational pull exerted on the otolith membrane bends the cilia of hair cells. Otolith organs detect change in the head position and linear acceleration. With a change in the head position, like tilting of head, change occurs in the direction of the gravitational pull exerted on its otolith membrane. This results in gravitational movement of otolith membrane. As cilia are entrenched in otolith membrane, head tilt bends the cilia of some of the hair cells towards the kinocilium. This increases the action potential frequency in 8th nerve. Action potential frequency in 8th nerve increases. Saccule detects linear acceleration in vertical direction. Example, experiencing acceleration while using the lift, and utricle detects the linear acceleration in horizontal direction. Example, experiencing acceleration in a car when car starts moving. Okay, so this is how the hair cells are stimulated, otolith hair cells. So when there is a head tilt, there will be the distortion of the hair cell that is going to excite the cell, and during the linear acceleration, because of the inertia of the otolith organ, the hair cells are distorted, and that is going to stimulate the hair cell. Semicircular canals. In semicircular canals, hair cells are located in ampulla, the dilated end of each canal. The ampullae open into the utricle. Each ampulla contains crista terminalis located on the pendular hillock, as it appears like a small hill. Hair cells are present in the crista ampullaris along with supporting cells. The cilia of the ampullary hair cells are embedded in a gelatinous mass known as scapula, which is an encapsulated inverted cup-like structure that forms a watertight space between canal and the utricle. However, gelatinous mass of cupula does not contain otoliths. Thus, specific gravity of cupular fluid is same as that of endolymph. So this is the structure of crista ampullaris. You can see the hair arrangement of the hair cells, uh, the hairs of which they are embedded in the cupula. And the hair cells here in the cupula, they are exposed to the endolymph, and the hair cells, they also contain the kinocilium and the stercocilia. The cupula extends from crista to the top of ampulla and moves back and forth with the movement of the fluid in the canal. When head rotates to one side, the canals being part of the head automatically rotate to that side. However, the endolymph in the canals, due to its natural inertia of gelatinous fluid, does not move immediately for about 20 seconds. Therefore, initially endolymph lags behind, uh, as if the endolymph moves in an opposite direction to that of the direction of canal movement. Thus, if the head rotates to the right, the initial 20 seconds endolymph practically rotates to the left. Cupullar fluid, having the same specific gravity as that of endolymph, moves along with the direction of the endolymph. The cupullar deflection bends the cilia of the hair cells. In ampulla, the kinocilia are located towards the utricle. Therefore, displacement of cupula towards utricle bends cilia towards the kinocilium and stimulates hair cells. Thus, the head rotation is detected in first 20 seconds. After the initial lag period, the movement of the endolymph equalizes with the movement of the canal. Therefore, the rotation is not detected after initial phase. When the head rotation stops, the endolymph in the canal continues to move to the same direction for about 20 seconds, which causes the deflection of the cupula away from the utricle, and hair cells are inhibited. However, the arrangement of hair cell in the canals of both the ears is such that the beginning of rotation is detected by the hair cells in the ear towards which the rotation takes place, and termination of the rotation detected by the hair cells in the opposite ear. Okay. So this is how uh, the crista ampullaris is going to behave when at it is rest, and when it is there is angular rotation to the left, the endolymph is going to move to the opposite side, that is to the right side, and which is going to cause the distortion of the hair cells. Okay, so this is when uh, the normal semicircular canals, when head rotation starts. Okay, so there will be the movement of endolymph in opposite direction, and there will be increased discharge from the right semicircular canal, and when the rotation continues, so there won't be movement of the relative movement of endolymph uh, with a sensory organ, and there will be no extra discharge. But when the rotation stops, the endolymph is going to move in opposite direction, which is going to lead to hyperpolarization and leads to reduced discharge from right ear, whereas opposite thing happens in the uh, opposite semicircular canal. Vestibular pathways. Vestibular information is conveyed to the CNS via vestibular division of 8th cranial nerve. The cell bodies of afferent fibers are located in the Scarpa's ganglion, that is vestibular ganglion. These neurons are bipolar neurons. The central axons travel in the 8th cranial nerve, which enter in the brain at the level of pons and terminate in vestibular nuclei in the brain stem. There are 4 vestibular nuclei: lateral, medial, superior, and inferior. The lateral vestibular nucleus receives fibers mainly from the utricle, cerebellum, and spinal cord. The efferent fibers from the lateral vestibular spinal tract that facilitates the extensor group of motor neurons supplying anti-gravity muscles. The medial and superior vestibular nuclei, these two nuclei receive fibers from the otolith organs, semicircular canals, and cerebellum. Efferent fibers from medial nucleus to form medial vestibular spinal tract. The fibers from superior nucleus project to the vestibulocerebellum and oculomotor nerve nucleus. Inferior vestibular nucleus receives fibers from the cerebellum and project to the cervical spinal cord. Thus, vestibular apparatus through vestibular nerve and nuclei conveys information to three major structures, that is cerebellum, oculomotor nuclei and spinal cord motor neurons. Through cerebellar connections, it maintains equilibrium and coordinates postural movements. Through the oculomotor nucleus, it regulates eye movements and coordination uh, and coordinated with the body movements. Through the spinal motor neurons, it controls postural reflexes, especially by controlling the postural muscles of head, neck. Vestibular apparatus maintains the postural stability during the movement. So this is the vestibular pathway. The different sensation from semicircular canals, saccules, utricles, they go to the vestibular ganglion through the vestibular ganglion or Scarpa's ganglion. Then they will form vestibular nerve. Then they are going to relay into the vestibular nucleus. Okay. From there, the efferent can, through the vestibular oculomotor nerve, goes to the extraocular muscles for the vestibulo-ocular reflexes. Then the information goes to the cerebellum, and some of the information that goes to the spinal cord to the skeletal muscles for the maintenance of muscle tone. Vestibular reflexes. Vestibular reflexes are broadly divided into two categories, postural and visual reflexes. The activation of postural reflex maintains balance and equilibrium during movement to provide postural stability for movement, and activation of visual reflex provides stability of visual images in spite of bodily movements. The postural reflexes activated by vestibular apparatus are tonic labyrinthine reflex, labyrinthine writing reflex, and vestibulocolic reflex. That is, the contraction of the head and neck muscle in response to vestibular stimulation provides a stability of head position during the movement. Vestibulo-ocular reflexes. This reflex is vestibulo-ocular reflex. When head rotates, the VOR helps in the fixed vision on the same object in spite of the rotation of the head. The visual fixation is an important component of postural regulation. When head rotates, eyes move in an opposite direction so that the visual image remains constant. However, when rotation exceeds more 60 degree, the rapid movement of the eyes occurs in the same direction that of the head rotation. So this rapid movement leads to visual fixation on a new object. Then again, eyes move slowly in opposite direction. So this slow movement of eyeballs alternating with a rapid movement in the opposite direction gives rise to nystagmus. The slow component of this reflex is vestibular component, whereas the rapid movement is integrated in the superior colliculi and prepontine reticular formation. Pathway for visual ocular reflexes. Vestibular ocular reflex. The first order neurons start from vestibular apparatus and terminate in the vestibular nuclei in brainstem. The second order neurons project from vestibular nuclei to the oculomotor nerve nucleus, and the third order neurons originate from oculomotor nucleus to innervate extraocular muscles. The latency of this reflex is about 10 milliseconds. So these are the functions of summary otolith organs and semicircular canals. Through the vestibular nucleus, they're going to reach to the vestibular nuclei, and through the cerebellum, they're going to control the balance. Through the oculomotor nuclei, they bring visual reflexes, and through the spinal motor neurons, they help in the maintenance of the postural reflex. Some of the applied aspects. Importance of vestibular function is not realized unless there is a vestibular dysfunction. Motion sickness. Motion sickness occurs in a susceptible individuals due to the overstimulation of vestibular apparatus. Very often it is encountered while traveling in a ship or fast moving vehicles like bus. It heralds with nausea and palpitation and culminates with sweating, dizziness and vomiting. However, repeated vomiting occurs if person continues the journey. Antiemetics may give mild relief. Closing the eyes help in the reducing the symptoms. In severe cases, labyrinthectomy or ablation of the vestibulocerebellum is performed to cure the disease. Vertigo is defined as illusion of motion, usually rotation when actually no rotation is occurring. The vestibular system that senses the position and rotation of head if becomes abnormal, vertigo occurs. So it occurs in following conditions. Like in physiological vertigo, usual example in motion sickness. Uh, However, the motion sickness is severe form is pathological. Then central positional vertigo, lesion of the 8th cranial nerve and vertebrovascular insufficiency are the usual cause. Peripheral or labyrinthine vertigo. This occurs due to the disease of vestibular apparatus like in case of Meniere's disease. Benign positional vertigo, it happens by assuming a particular position of the head and usually occurs in elderly. Coming to the vestibular function test, the clinical test, assessment of postural balance test tests vestibular functions. For example, ask the subject to walk on a straight line or stand erect with feet together but with eyes open. With the eyes closed, the test is Romberg sign that assesses the postural imbalance other than cerebellar and vestibular origin. Then rotational stimulation, that is post rotated nystagmus. Vestibular function can be produced by rotating the patient in a Berenice chair. The subject sits on the chair with has a head bent 30-degree forward as the horizontal semicircular canals remain horizontal in this position and can be stimulated most effectively by rotation around the vertical axis. Subject's eyes are closed or he uses Fresnel lenses to avoid the effect of optokinetic nystagmus. The chair is revolved at the rate of 1 revolution per 2 seconds for 10 revolutions and then suddenly stopped. If the vestibular function is normal, nystagmus will be observed for 20 to 30 seconds. Caloric stimulation, that is caloric nystagmus. Vestibular stimulation can be produced by irrigating ear canal alternatively with warm and cold saline. The test is based on Berenice's theory that warming the endolymph decreases its specific gravity, which sets up convection currents that produce movement of endolymph as occurs during angular acceleration. The patient is kept in a supine position with his head bent forward by 30 degree. In this position, the horizontal uh, semicircular canals remain in vertical position and the external auditory canal is syringed with gently first with the cold water that is of 30 degrees Celsius and for 30 seconds and then with the warm water 44 degree with a gap of 5 seconds between them. The procedure leads to nystagmus if semicircular canals functions is normal. With a warm water, nystagmus occurs to the irrigated site for about 90 seconds. Then electronystagmography, this provides a more accurate assessment for labyrinthine functions. So this is in brief about the vestibular apparatus, its function and some of the vestibular function tests. Thank you.